All right, so when we go into the lab today, it's going to be very similar to the other day. You're going to grab a microscope, okay, and there's going to be some slides that you're going to need to look at, okay? The main two slides are going to be the monocot dicot cross section that we talked about yesterday. Okay, I'll go over that again, and the generalized animal cell. Okay, those are going to be the two most important. Those are the ones I'm going to want lab diagrams of. Okay? You don't need to do a lab diagram, labeled lab diagram of the blood cell, right? Because you can't really see anything on the human blood anyway, other than the cells themselves. All right, so I want to see 3 lab diagrams, but only 2 of them need to be labeled. Okay? All right. So the problem we're investigating has to do with the cell theory, specifically the second point of the cell theory. What do cells do? Cells carry out the basic functions of an organism. Okay? In order to do this, they must have certain structures in their cells that carry out those basic functions. So the purpose of our lab is to figure out which structures are essential for cells to carry out their basic functions. So we're going to look at 2 different kinds of cells and see what they have in them. Okay? Hopefully, we're going to see a lot of commonalities. Okay? That is a lot of structures in both the plant and the animal cell. And we're probably going to see some differences as well. And those differences are probably going to be attributable to the fact that they use different modes of nutrition, something we talked about the other day as well. Okay? A plant is a photoautotroph, an animal is a chemoheterotroph. Okay? So there's going to be some structural differences because of that we'll be able to see. Okay? All right. So for our design, okay, we're looking at different cells again. Okay, we're looking at different cells again, okay, and we're looking at a plant and an animal. We need to look at different cells because we need to verify that cells have common structures that help them carry out their basic functions. Okay? So that should help you with both the manipulated and the responding variables. Looking at different cells, and I told you why. What we're looking for in each cell would kind of be your responding variable. Okay, and I'm going to give you one of the control variables. Okay, your first control variable is all the cells we're looking at are eukaryotic. Okay, that is spelt like this. We talked about it yesterday. Eukaryotic cells are cells that have all those compartments, okay, or they have compartmentalization. It does us no good to look at a bacteria because A, they're really small, and B, they don't have any of these compartments. Okay, so it wouldn't help us to confirm our hypothesis. Okay, you need to come up with 2 other control variables, things that need to be the same each time you look at a new cell under the microscope. Okay, we're going to be viewing both of these on the highest power possible. Okay, you'll see the most detail at that level. For our hypothesis, okay, the if part has to deal with that. Okay? It has to deal with the second point of the cell theory. If that's what cells do, okay, that's what we're working on here. Remember that we're trying to figure out what structures are needed for cells to carry out their basic functions. So the premise we're working on, that's what goes in the if part, is that cells do just that. They carry out the basic functions of the organism. Okay, and part, as always, describes the experiment. Eukaryotic cells are being viewed under a microscope at high power to discover their structures. I don't know, something. Okay, that's what we're doing. Okay, just describe the lab in brief. Okay. So we said the premise here is cells carry out the basic functions. What we're trying to figure out is what structures do they need. So the then part should tell me what structures you expect to see in each cell and why. I'm not expecting you to write down all 13 things that are on your on your cell diagram from class the other day, because you're not going to see all of those. Okay, you're not going to see ribosomes. Okay, but I do expect here that you would list some of the things you would expect to see in these cells and why. Okay, so you actually have to list a few organelles here. Okay, in the then part. Any questions so far? So the procedure, pretty straightforward. Okay, I'll have the cell trays, or the slide trays set out. Okay, on the front table, just like I did for the microscopy lab. Come up and get one slide at a time. Okay, don't take multiple slides back to your table, because inevitably, while people are looking through the microscope, they don't know what their hands are doing, and slides get knocked off the table all the time. Okay, so we don't need to have any more slides at our table than the one that's in the microscope. Okay, so just come up, take them as you need them. Okay, get your good pictures. Okay, of the monocot, dicot cross section of the animal, generalized animal cell, and a picture of the blood. Okay, that you can use. Okay, for your lab diagrams, and then that'll be it. Once we've got those pictures, then we'll come back here and you'll have time to work on your analysis and conclusion and all of that. So, talking about the analysis. Okay, like I said, there's one analysis question here. Based on your knowledge of cell structure and your observations of the cells in this lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Explain your reasoning. All right, so basically what you're doing here is you're telling me these are the organelles I saw in all the cells, and here's why I saw them, why they're important, why they help carry out the basic functions. And then secondly, here's an organelle I only saw in the plant cell. Here's why I only saw it in the plant cell. Here's an organelle I saw only in the animal cell, and here's why I only saw it in the animal cell. Okay, that's what I'm looking for. That's going to be a paragraph if you do it right. Okay, I'm thinking like the bare minimum here is probably 4 sentences. That would be the bare minimum to do a good job of that question. Okay, conclusion, copy paste your hypothesis and then explain using your observations. Okay, what things did you see? What were common to all the cells? Does that help to confirm that basic functions are carried out by these particular organelles like you stated in your hypothesis? Okay, evaluation, okay, your sources of error, right? What are some things that could be done better in the future to make it easier to find all this stuff? Okay, so quick reminder here of the things you're going to be looking at. Okay, so you're going to be looking at a monocot, dicot cross section. Okay, when you get that slide, okay, it's going to look like this. There's going to be 2 little wedges under the cover slip. Okay, you want to be looking at, I believe, the bottom wedge, not the top wedge. Okay, and you want to look at this part here. So here's the wedge. You want to look at this part here. Don't look at the middle. Okay, because in the middle is where all the veins and tubes come together in the leaf, and all you're going to see are a bunch of empty circles. okay? Because you're gonna be looking this way into a tube, okay? So it'd be like looking down a pipe. There's nothing to see there, okay? What we wanna look at is this part of the leaf. That's where we're gonna see this, okay? This is that part of the leaf. And we wanna look at specifically the cells near the top. That would be in this layer here, okay? That's where our best chance of finding a really good plant cell are gonna be found, okay? This is what we call the Palisade layer, okay? Palisade is like a rock formation with columns of rock, okay? And that's what these are. They're columns of cells, okay? All stacked really close together, right? So we want to look for an individual cell, okay, and get the best picture we can of that, and then you're going to label it. Now, if you need to label more than one cell to label all the structures you can see, that's fine, okay? You're labeling a diagram, okay, but you want to try your best to get one really good cell if you can. When you look at the animal cell, okay, you're going to see this, okay, we talked about this yesterday, all right? If I'm looking at an individual cell here, okay, that would be like right here, that would be one cell, okay, right there, right? And so you can do your labeling. There's going to be lots of cells. Every one of these little purple circles, okay, is a nucleus. And if you can see the nucleus, can you see the nuclear membrane? Yeah, you can, okay? If you know where the nucleus begins and ends, then you can see the nuclear membrane as well, just to help you out with your labeling, okay? And then, obviously, you can also see the nucleolus inside of each one of these. And if you look really carefully, come on, get bigger. Okay, on a lot of these nuclei, you can see on one edge that it's a little rougher on one side than the other, okay? That's usually the rough ER, okay? If you're looking for things to label, okay? That's stuff that you can see for sure, okay? You can also see cytoplasm, and you can see cell membrane, and all that kind of stuff. So there should be plenty for you to label. Just don't make stuff up, okay? If you have ribosomes labeled on your diagram, I'm gonna call you on that. You didn't see them. They're too small, okay? No matter how big I make that diagram, I'm not seeing ribosomes with a light microscope. Okay. It's possible you might get a Golgi, you might be able to see some lysosomes, okay, stuff like that, right? But you're not going to see the really, really, really small stuff. All right, questions on what we're doing when we get over there? Okay. So we're going to quietly make our way over to the bio lab, okay? Take your phone with you, right, so that you can get the pictures you need.